Okay. So we are Jenna Bath and Rupert Pepra. We're both from Damon University and are in the Department of Natural Science. We chose to do our poster on the lipid percentage effect on pollutant absorption in fish. So a small introduction. We took this fish samples from U.S. domestic waters. We tested the effect of lipid percentages in fish when absorbing pollutants. We focused more on PFAs, or PFA absorption and pollutant absorption because we, as a society, as a population, have been more and more increasingly affected by pollutants getting into food, water, contaminating everything. And one of the more important factors is that it's also impacting infant neurological development if it gets into their food sources.
So basically, like Jenna was saying earlier, our study focuses on understanding whether the amount of lipids in fish influences how much pollution they absorb, specifically PFAs and PCBs. We examined three common U.S. freshwater species, like Jenna said earlier, the largemouth bass, the smallmouth bass, and then the Chanel catfish. These species were chosen because they are widespread and appear frequently in national toxin databases. Our goal was to determine whether fish with higher lipid percentages absorb more pollutants, as is commonly assumed. Methods. We collected data for 50 fish samples using the EPA fish toxin database, which includes pollutant concentrations in domestic U.S. waters. We also used Model My Watershed to collect environmental data, including livestock frequency around sampling sites. Basically, all statistical analysis were performed in SPSS28 using the t-test, the linear regression, the ANOVA, with a significance level of 0.05. The main analysis tested whether lipid percentages predict pollutant absorption. Results. So looking at figure 1, the mean lipid percent for samples collected from non-urban and urban sites shows no statistical significant differences between site types. When you come down to figure 2, that is the scatter plot showing the regression of the trend line when referring to the association between lipid percentage and PFA levels, having an R2 value of 0.0444 and one major outlier. When you come down to figure 3, that is the mean PCB concentrations among 3 fishes. The error bars represent the standard errors of the mean, and they share the letter A because there are no significant differences. Basically, across all these 3 species, lipid percentage did not significantly influence the PFAs or PCBs. Our regression analysis produced an R2 value of 0.0444, meaning lipid levels explain only about 4% of the variation in pollutant concentration, essentially showing no meaningful correlation, like I said earlier. We also found no significant differences in lipid percentages between urban and non-urban sites, and no significant differences in PCB levels across species. One noticeable feature in the data was a major outlier, but even with that point, the overall trend remained weak. Conclusion. Basically, our overall results do not show that lipid percentage has a statistical significant effect on PFA and PCB absorption in the 3 selected fish species. While this contradicts some expectations from past research, it highlights how pollutant absorption may depend on factors other than lipid levels, such as water chemistry, feeding behavior, or local contamination patterns. Although the results did not show a correlation or relationship between the lipid levels and pollutant absorption, it can help further studies to determine pollution absorption in native wildlife. Basically, what I'm trying to say is that even though our findings doesn't review a correlation, They provide useful information for future ecological use, for toxicology studies, looking at the pollution exposure in wildlife. Thank you.